So first of all, I'm going to be talking, following on from Sue's results and from Sue's talk about this vaccine effectiveness study. And thank you very much to Sue for the opportunity to be part of it and to be here today. I'm going to go quite quickly, just 15 minutes. We're at a fairly early stage of this study, so really I'm going to introduce the study team and the research question, and I'm going to talk about the methodological challenges and talk about what's our plan for addressing them, but also to talk about the strengths that we have in the data. So this paper is to estimate the effectiveness of COVID-19 vaccination on disease incidence and severity. And this is a collaboration between ESR and the University of Otago. It's lovely to be all working together. Sue is the study PI, as she mentioned. I'm the lead on this paper. And we've got the Otago team that's going to be managing the analysis. And the ESR team is providing us with the data. And also with they're contributing in terms of their very deep understanding of the cohort and the data. And that way of collaborating is very exciting. Lots of methodological challenges, and it's useful to think of them in terms of sources of error in this analysis. We've got systematic error, which is bias, and we've got random error, which arises from random variation. I won't talk through all of this slide, but you can see there are a lot of things to consider. In particular, we've got selection effects, so in any cohort or any population, any time period is going to induce a selection effect that you have to think about. And for measurement error, a really important one for COVID is, of course, case ascertainment. We know that COVID is usually under-ascertained, and so part of the value of this kind of cohort study is we've got a better opportunity to ascertain cases. In terms of confounding, I'll come to that in a minute, lots of sources of confounding. And random error comes from cohort size, but also just because we have, it's not simple binary in terms of our exposure, vaccinated or not vaccinated, but different dosages and some time factors also. So there's some methodological challenges here and some design decisions to be made at the analysis stage. The analysis plan is fairly straightforward. What we want to do is develop a causal framework, our causal thinking about how we think this is going to work, and then implement that causal model in the data as a statistical analysis. And along the way, we'll do some descriptive analyses. I'll show you some of those initial ones, and you've seen some from Sue. And we'll do our estimate of effect, and then go back to those sources of bias and do some bias analyses to see how sensitive our estimate is to the decisions that we've made. And finally, I hope we can contextualize this study by through dialogue with other groups that are doing VE studies in the whole New Zealand population, and that's going to be a very interesting conversation. So starting with the causal graph, and this is actually a simplified version, but suffice to say there are a lot of sources of confounding here, so a lot of backdoor pathways that we're going to need to close off to get our estimate of effect. We're very, very fortunate to have this wonderful cohort to study. This is a very well-designed community cohort with very well-motivated participants, and what that means is we have very high-quality measures in this study. We've got detailed measures, and I'm sure everybody here is familiar with the sort of measures we have at our disposal. In particular, we've got a very good coverage of those confounders that I showed you in this previous slide. And we've got good case ascertainment and opportunities to link the data to other sources, other data sources, and that gives us a lot of knowledge and context to work with. This is a perspective unit, longitudinal design. It was such an advantage because when the Omicron outbreak started to develop in New Zealand, this cohort was already set up, the data processes were set up, the data collection processes were set up, and it was ready to go. So this is, we've heard from others others today, this is a tremendous example of why it's important to have these longitudinal cohorts in place in the event of something unexpected, like a pandemic. We've got some individual and household level data in the Wild Kiwis cohort, and that means that's really good at capturing how infectious diseases really work. And what I'm hoping is Naira will be talking about her study of household transmission. When we've got the VE results from our study and the household transmission results, we can put those together. We can start making really good evidence-informed recommendations for vaccine policy. And finally, we've got international comparability, as we've heard, but also local relevance. This is a cohort for New Zealand. It captures New Zealand's unique situation. In particular, that when the Omicron variant started circulating in New Zealand, most of the population at that point was naive to COVID-19. So we don't have to, it's one thing we don't have to worry about is previous infection as a complicating factor in the VE estimate. So very straightforward analytical approach. Important to think about the time factors because they vary over time. We've got eligibility, case numbers, variants, vaccine waning, all of those things change over time and we need to capture that. A Cox proportional hazard model seems to be the most appropriate for the main analysis, and that's been done very successfully outside New Zealand and other VE studies. And of course, one of the things we will be thinking about very carefully is how to adjust for multiple sources of confounding, as I mentioned. Sue has shown you some of the descriptive results, and I'm just going to show you a little selection, very high level, just to show you the kind of digging in the data that we now need to do at this stage. You do develop your causal model and go into the statistical model from that, but in reality, there's a little bit of toing and froing to understand what's in the data, which can determine what is feasible to do in the analysis. And on this slide, I just wanted to make one point here, which is we've got our exposure, which is vaccination, actually has 5 levels. And what that means is that some of those cells there have very few observations, and we need to take that into account in terms of collapsing down some of the strata that we've got. I also want to make a point on that slide, relatively low number of observations for Pacific people, so we don't expect to be able to say very, very much about those populations. In this one, which is just the percentages from the previous one, really the main point to make here is that this is a highly vaccinated cohort. And when we look at the predictors and the confounders, very useful to have a look at what you find in simple descriptive results, just to see if it matches, if it makes sense. Does it make sense in terms of how you think things work, how your causal model is organized? And we can see that it's showing the patterns that we would expect to see. It does make sense, and that gives us a confidence in terms of what we're going to adjust for. So, in conclusion, we've got the SHIVERS data that present an opportunity for a VE study that is methodologically rigorous in terms of our estimate of effect. We've got lots of opportunities to address those sources of bias. We can identify some strengths and gaps in the national data, and that's going to be a very useful comparison to make. And most of all, it's New Zealand aligned. It's providing appropriate and very location-specific evidence for action. Next steps, we've got a few more analysis design decisions, and so one of those, as I mentioned, is how do we simplify our strata without losing information or distorting the relationships in the data? And secondly, how exactly to adjust for confounding. We could do a propensity weighting approach, or we could do a simple adjustment, and we'll have a think about which would be the best approach for us in this study. And with that, I think I'll stop. And thank you very much to Sue for the opportunity.